Yes. Oh, yes. Hi, thank you very much for inviting me to such a fun seminar straight from Europe. I like such an interdisciplinary event to learn and tell what we are doing and interest each other. So I know that it should be a tough time in America. So I hope maybe I can see you in the recorded video later as well. My name is Saitou. I am an assistant professor at the Norwegian University of Life Sciences. So today, I'd like to tell you a little bit about how genetic difference tell us about our evolutionary history. Yep. My primary motivation in research is to find something interesting. So while I know that others do science to develop something useful or helpful, I do appreciate technology and would be very happy if my research can be ended up helping somebody else, of course. Thanks to the technological breakthroughs, now we can read genome sequences at a relatively small cost. For example, if you want to sequence a genome of one person or your dog or cat, it will only cost $1,000 nowadays, while it was $100 million in 20 years ago. So thankfully, more and more genomic information is piling up, published, and ready for research. By analyzing these datasets, we can gain insight into medical genomics to find the disease-causing genes, insight into agriculture, livestock, and aquaculture by associating genes and favorable traits, such as fast growth and richer nutrition, and also insight into our evolutionary history. I have been investigating the evolution of various species from fish, birds, to mammals using genomics. Today, I will talk about the work I have done during my postdoc at Buffalo, US, which was published last year about human population differentiation. So how can DNA tell us about our evolutionary history and individual difference? DNA is a long chain of nucleotides, and the combination of sequences can determine how to structure our body and how to regulate our body function at the molecular and cellular chemical levels dynamically. When DNA is copied, for example, at the cell division, sometimes DNA replication system can make a mistake and insert the wrong nucleotide. Or sometimes the system makes long genetic deletion or insertion, or more complicated errors. The human genome consists of over 3 billion base pairs. But at each generation, about 30 mutations can be introduced. And even twin siblings have several differences each other in nucleotide by mutation. So most mutations are neutral and function similarly to the original type. But some mutations can be deleterious, such as causing a disease by inducing the malfunction of a gene. And sometimes mutation can be advantageous by introducing a new, better function of a gene. So the accumulation of mutations, including random, deleterious, and advantageous mutations, can form the differences between individuals, populations, and species in the long run. Let's assume that each dot here is one individual in the population. And the left is a past population, and the right is the present population that we can observe. So the majority of the past population has the blue type of gene, but we see that one individual has a pink type of gene caused by mutation. So if the pink individual can reproduce more offspring than others, the pink gene could be inherited more than the blue gene. And after generations and generations, we can observe more pink genes in the population. This can happen for neutral mutations by chance, but if the pink gene is more advantageous than the blue type, the pink type can increase its frequency in the more rapidly and at more higher probability. So by using statistical methods and simulation analysis in the population level genome sequence data, we can identify the potentially advantageous genetic mutations. So as we have reviewed so far, genetic mutations can tell us about evolution and diversity of species and population. However, the most genomic studies so far focus on only the simplest type of mutation among the many. So here are the various types of genomic structure variants, including genetic deletion or insertion or duplication, inversion, and copy number variation. The other type is the single nucleotide variant, which just changes one nucleotide. Since the size of genomic structure variant is much larger than single nucleotide variants, so they have more impact, but they are more difficult and more costly to precisely identify and analyze. So the majority of genomic structure variants were left uninvestigated well. So as you may expect, our aim of that research was to understand the evolutionary history of the underappreciated types of mutation, genomic structure variation in humans in this study. So, to do so, we first wanted to determine the geographically differentiated structure variants in human diverse populations, and to investigate if those variants are associated with any non-human trait variation. So first, we used the public whole genome dataset of thousands of human individuals from 26 populations from various geographical regions. In those whole genome sequence dataset, there are 57K genomic structure variants were identified. So to quantify the unusualness of the geographical distribution of each variant, we established a matrix-based measure named the measure of unusualness. Then, we calculated the unusualness of each gene by comparing each pair of the population and to see how unusual the geographical distribution is for each gene. Then we took the top 1% unusual genomic variants for further analysis, assuming that this might be under geography-specific evolution. So here is one example of unusually distributed genetic variation we identified. A partial deletion of DAP, death-associated protein gene, which is associated with cell death. So we see that the deletion, partial deletion type, is common in Africa, the blue one. And the other one, non-deleted type, is common in Europe and Asia. The pale blue means that the percentage of people with both types, means that who inherited the deleted type from one parent and non-deleted type from the other parent. So they have both. There is also another interesting observation on this gene. So the deletion was observed in some modern humans, as we saw, but also in the Denisovan genome, which is extinct archaic human species like Neanderthals. So here are the genome data of 2 modern humans and Neanderthal and Denisovan. So we can see the gray bars, which are the fragmented and sequenced DNA information. Here are, but we also see 2 white blanks here and here in 2 individuals, one modern human and one Denisovan genome, which means that this part of DAP gene was deleted only in those people. So then what is the effect of this deletion? How can we know the effect of genetic variants of interest on organismal traits? So there is another type of study, the genome-wide association study. which tries to identify the genetic variants that are associated with a particular trait. In that kind of study, for example, researchers recruit multiple groups of people, say people with dark eyes and people with blue eyes, then compare the genome of two groups and identify different variants or identify the genomic variants which show extremely different ratios between the two groups. So there are some databases which aggregated that kind of associations. And we used the result of these genome-wide association studies to estimate the effect of highly differentiated variants. So previously we have identified unusually distributed genomic variants from the whole genome sequence dataset of 26 populations. And also we used another reported associations between variants and the traits. However, there's a major problem again. So most of the studies, that kind of studies, it only focuses on the simplest type of variants because it's cheaper and easier, single-nucleotide variants and not structure variants. So we also established a statistical method to estimate the effect of the effect of structure variants. Using the neighboring single-nucleotide variants, which are physically tightly linked with and behave similarly to the target structure variants. So by using the neighboring variants, we can estimate the effect of structure variants of interest on humans or other individuals. So using that one here is a list of genomic structure variants which showed marked geographical distribution and were associated with some putative effect. The green number is the observed frequency of each variant in Africa, East Asia, and Europe. And the trait includes vision, height, or drink temperature preference, hair type, morning personality, etc. So this color indicates the differentiation between subpopulations. And the bottom is the variant we looked into previously, the one in common in Africa, and it was shared with the Denison one. So this variant is associated with white blood cell numbers counts. So it might be related to immune function to fight against some pathogens there. However, the limitation of the current human genomics in this study and also in general is that the majority of genome studies are focused on European ancestry individuals. And there is little available data about non-European genome or non-European populations. So for example, we found many African-specific or South Asian-specific variants. But the putative function of these non-European specific variants were not well investigated and reported. So I hope the diversity of human genome will be appreciated and more diverse genomes will be investigated. And not only for science, but for the equality of gain, broader benefits from the insights. So this research was initiated at the University of Buffalo while I was doing a postdoc there and has been published in a journal, Molecular Biology of Evolution, recently. Thank you very much for the opportunity to present this work.
Absolutely, thank you for a great talk. That's really interesting that you're able to parse through the data set like that. So just generally, how do you?
Yep.
So how would you take this information and sort of draw further conclusions from it? So you showed, like a higher variability. So what does that tell you in terms of like what would a next step be to try and understand it better?
Okay, so we know the genetic variants and the macro phenotype. So it would be great if we can test how it's beneficial at the cellular level. For example, it makes some different types of protein, or it's like elongate cell longevity, for example. At the that would be great.
Okay. So at that point, once you identify a specific variant, then somebody would be able to go in and say, okay, we know this variant shows up a lot. What is it doing? Where does it come from? Is that kind of how it would go, or?
So in ideal cases, if those putative variants are easy to test, one thing is to make a genetically modified mice and raise them in a different conditions or different nutrition, then test. One mutant type is more beneficial.
Okay, that makes sense. So I'll ask one last question. What sort of not limitations necessarily, but what do you have to watch out for when you're doing this kind of analysis with the data? So that you don't get like false positives, or
Yeah. The one major thing is a highly European bias. Data set itself. There should be a lot of layer of biases. For example, I think these data sets are mainly for medical research purposes. So I think non-disease associated traits are less investigated, maybe.
Okay. Yeah, I guess that makes sense. You would have to be sure that it's a representative data set for what you wanted to actually explore. Okay. Well, fantastic. Thank you for sharing this work with us.
Thank you very much.
Appreciate it.
Yeah.
And
if
I can get you to hang around for just 1 second, we'll talk very briefly. So thank you to everybody for coming out.